All right, everyone, welcome to our second session of Student Talks. Thank you for everyone who has come to support hearing our local student speakers. We've got another 5 wonderful talks from students who are currently getting a master's degree or a PhD, or those who have just graduated. So our first talk today is going to be Stephanie Calloway. Stephanie just graduated from Cal Poly San Luis Obispo, where she studied the island mallow, and she just recently started her absolute dream job as a botanist in the El Dorado forest. So please, let's welcome Stephanie up to the stage. Thank you, Natalie. Can everybody hear me? Good. All right. Thank you for being here today. I'm so excited to share my research with you on the Northern Island mallow. The Northern Island mallow is a perennial shrub in the Malvaceae, and as you can see, it just has these beautiful flowers that cover this shrub. It occurs on the Channel Islands located off the coast of Southern California, and these islands are considered biodiversity hotspots. There's over 240 endemic plant species on the islands. But unfortunately, decades of grazing by introduced herbivores like these sheep you see here completely decimated island biodiversity. And these herbivores have been removed from most of the islands for about 20 years now, and recovery efforts are underway across all the islands, but unfortunately, many species have still failed to recover, including the Northern Island mallow. So specifically, the Northern Island mallow is native to 2 of California's northern Channel Islands that are managed by the National Park Service. So San Miguel Island, where the island mallow is known from about 150 individuals, and Anacapa Island, which is the specific focus of my research that we'll zoom into now. Anacapa can be separated into 3 islets that are completely separated by the ocean. So West, Middle, and East Anacapa. And natural populations of the Northern Island mallow are only known from West and Middle Anacapa, as you can see from those hollow green circles. Fortunately, though, they have been presumed extirpated on Anacapa since the 1990s, unfortunately. But prior to extirpation, seeds were collected from the last remaining plants and used to create a new population on East Anacapa Island, those green dots, the filled green dots you see over there. And that is the specific focus of my research, is that reintroduced population. And at that reintroduced population, there are about 850 individuals that were planted starting in 2016. And zooming into that restoration site more, that whole green blob is the restoration site that I'm talking about for my research. And those green hatch marks within that green blob represent Northern Island mallow at that site. And in addition to all of the mallow that were planted, the Park Service planted about 8,000 additional coastal scrub species transplants within that site as well. And this is what the site looked like in 2016, just before restoration. As you can see, it's really covered in invasive plants. Almost every plant you can see in that picture is an invasive species, and that's really indicative of what most of the island looks like, where over half of the cover is an invasive species. And then just a few years after restoration, this is what the site looked like. So just an amazing transformation. I didn't do it. I just worked there. But native plant cover and richness had both increased by 6 times, and island mallow accounted for about 15% of the cover within that site. And in some areas now, island mallow even grows over 8 feet tall, and it can produce thousands of fruits per plant. So it sounds like a pretty awesome success story so far. But the problem, unfortunately, after restoration, there's been almost no new recruits observed in the restoration site. So a recruit is simply a member of the next generation that has naturally germinated from the seed bank and grown to become a juvenile. So, these plants are prolific fruiters, and they have near 100% germination in the greenhouse. So what could be going on that is preventing recruitment? You would think, we'd walk through the restoration site and see these all over the place. Well, biologists have observed predation of flower buds, fruits, and flowers by the Anacapa deer mouse, which is a special deer mouse that only occurs on Anacapa Island. And it's also the island's only land mammal. So there's no foxes on this island like on some of the other islands. And you kind of walk around the site and you see all these just like piles of fruits and flowers all over the place. So it's very clear that they're eating these things off the mallow. So the National Park Service was interested to know if the deer mouse could be playing a role in limiting the recruitment of this rare species. So with this information, we can think about these problems in the context of malva's life history. So there are several things that can happen during these early life history stages that limit and will ultimately prevent mallow recruitment. And that's including things like seed and seedling predation. And based on the large-scale evidence of that seed predation that we're seeing and the total lack of seedlings within the site, we hypothesize that deer mice limit recruitment via seed and seedling predation. And to test this hypothesis, we performed experiments that excluded deer mice from each of these different life history stages, so reproductive adults, dispersed fruits, seedlings, and juveniles. And we either exposed these different life history stages to deer mice or protected them from mice. And we then asked, what is the probability that each of these different life history stages would transition to their next life history stage in the presence and absence of mice? And in each case, we predicted that the life history stages would have a greater chance of transitioning in the absence of mice. And I'm not going to be able to go into every single one of the experiments that I did, but I'm going to talk about the first couple briefly, sum them up, and then go into more detail with the seedling and juvenile life history stages. So for those first two stages, the fruits while they were still on the reproductive adult, and then dispersed fruits, when we exposed those fruits to deer mice, we found that essentially all fruits were either eaten by the deer mice while they were still on the plant before they had a chance to actually mature and become viable fruits. And those that didn't get eaten while they were on the plant got eaten after they dispersed, or were removed by deer mice after they dispersed. And that is compared to being protected from deer mice, where essentially all of the fruits transitioned to become viable fruits that could disperse and remain in the seed bank. So this seems like pretty strong evidence that deer mice are limiting recruitment during this life history stage. But at the same time, these experiments didn't really account for any seeds that are remaining in caches that are unused by deer mice. So what we really want to know is what happens during that next stage. Can they even germinate from the seed bank? So yeah, can our seedlings germinate and survive? So to answer this question, we measured the germination probability of seeds in the presence and absence of deer mice. And we predicted that seeds would have a greater probability of emerging to become seedlings in the absence of deer mice. So we randomly selected 30 adult mallow throughout the restoration site, and we replicated this experiment for 2 years in 2020 and 2021, and experiments were started before the rain events of each year. This is an example of what the setup looks like at a plant, and we put the plots kind of in the fog drip zone of the adult mallow because they have this really nice fog drip, and this is where we observe them germinating in the wild in places where they do germinate. For each location, we sifted the top couple of inches of soil. We sowed 15 seeds per plot, And this totaled in 1,350 seeds sown per year across those sites. Here's just an example of what that looks like at a plant, and then we returned every 1 to 3 weeks to record emergence and mortality. And here's our results. So we saw that seeds do have a greater probability of emerging in the absence of deer mice. So looking at 2021, treatment on the x-axis and the percent of emerged seedlings on the y-axis, we only see seedling emergence in the caged treatments, and this was still during a really bad drought year where we only got 3 inches of rain, which is just abysmal for Anacapa. And then really the same pattern in 2022, except we had over twice as much rain, so we got a lot more germination. So this, to me, is really strong evidence that deer mice can limit recruitment at this life history stage, but it still doesn't really get at some of those seeds that are escaping in those unused caches. Like, I think most of those are getting eaten, but you know, any of the seeds that got removed from that experiment could still be at play here. So for our next experiment, we wanted to look at seedlings that we had germinated and remove the cages from them and see, you know, what happened. Would the deer mice actually eat the seedlings? So we did this in 2021. After we did our germination experiment, we pulled cages from half of those seedlings, and we predicted that they'd have a greater probability of surviving and transitioning to juveniles in the absence of deer mice. And what we found, no graph yet, but exposure level was not a significant predictor of whether or not these seedlings survive. So they all pretty much died regardless of treatment, but we wanted to look at the way things died to further investigate this pattern. So here's a plot showing treatment on the x-axis and the proportion of seedlings on the y-axis, and then all of those different colored boxes represent different fates of the seedlings. So for exposed seedlings, 67% died from desiccation, 33% died from deer mouse predation, and then for protected seedlings, almost all of them died from desiccation, even though they were protected from mice. And then we just had one that survived. It's actually a lot bigger than this one now, and it's recruited to the life history stage and to its next life history stage, and to my knowledge, it's like still doing good. But yeah, kind of what this really shows is there's not really a true statistical difference that we saw in the way things died. Like, if you're a seedling, you're probably gonna die from desiccation. But at the same time, you know, the caveat here is that this is during a drought year. With more water, you know, deer mouse predation may become more important. But at least during these drought years, it seems like desiccation is a big deal. But at the same time, you know, your only chance of survival is being protected from deer mice. That's the only, you know, seedling that we saw survive. So if drought doesn't get you, the deer mice will. So in conclusion, our hypothesis that deer mice limit recruitment via seed and seedling predation is mostly supported, especially for the seed life history stage. During the seedling life history stage, drought and lack of water seems to be a little more important, but at the same time, the only chance you have of survival is being protected from deer mice. So what we do know is that malva needs continued active conservation on Anacapa in order to continue to persist out there. It's a short-lived plant, and the whole population is pretty equal age, so we could see, you know, all of these plants dying at once, and, you know, without some active work out there, it could disappear again. So some recommendations would be to regularly collect seeds and store them on site or in a seed bank. Could potentially cast seeds during El Nino years. If we know the deer mouse populations are low, the Park Service tracks their densities out there. And also, these plants are super easy to grow. They're an amazing restoration plant. It's really lucky that, like, they do have 100% germination in the greenhouse, and you can just easily grow them and go plant them. So with that, I want to say thank you to all my supporters and everyone who helped out on this project. And I'll take any questions if there's time.
We do have time for questions.
Hi, awesome talk. I love how on these islands all the rare organisms just eat each other, and you have to choose one to prioritize.
Right.
I'm just curious whether you think that at some point, if restoration on the island proceeds really well, like the predation, like just the amount of plants, like can we overwhelm the deer mice with seeds so that eventually this ongoing like planting of juveniles could cease?
Yeah, I think that's a great question. I think there's a lot of opportunity for like more research to get at that question, like through doing like a minimum viable like population analysis with all of these transition rates that you can get from like a big life history study. Right now, the island mallow these restoration sites kind of like around that part of the island are kind of the only show in town for a big part of the year, and like the rest of the island is really like invasive species. So it would be really interesting to see like seed preferences on other plants on like the rest of the island and how that interplays with what's going on in the restoration site. They're continuing to like, their plan is just to keep moving the restoration out too and making it bigger every year. So I think, the fact that they eat everything doesn't necessarily bode well for that, but yeah, I think just more research needs to be done to really know like how many plants we need for them to be able to like reproduce on their own.
Hi, I wanted to ask if there was any understanding of why the deer mice predate on the juvenile mallow.
My understanding from my observation out there is that it's a really dry island, and there's not very much water, and I think that the deer mice really just like anything that's like nice and juicy to eat. So like there's Dudley out there, and they'll just like eat all the Dudley leaves on like these rare Dudley, and it's kind of sad. And when they're eating the fruits from the adult plants, they're like these green, like delicious, moist fruits that they're not going to be able to like save in cache. So it seems like they're probably kind of eating them right away, and it might be a water thing, but I'm not really sure. Yeah, they'll actually kill adult plants sometimes by like gnawing at the bark and to get moisture and like girdling a stem.
Thanks.
Yeah.
Any other questions for Stephanie? We still have some time.
Hi, sorry. I'm curious, sometimes, in restoration we make these really open areas, and then the native herbivores like can cue in on, oh look, this looks great, I want to eat these plants. Have you thought about like the complexity of the surrounding vegetation? Like are these, I saw couple photos, but is it just like this nice now opened area without these invasive species, and they're just able to cue in on these plants? Or have you thought about that?
Yeah, there's like a true lack of invasive species in the site. It's pretty amazing. They continue to like actively manage for the invasive species in there. So it's really, really different than the surrounding vegetation, which, is mostly, it's like a crystalline ice plant and these like flats of invasive grasses. Yeah, I think it would be really interesting to like understand like, are like what else are they eating? Are like mice eating these other invasive species, and could it be like boosting their populations higher? Or like do they really just favor the island mallow more than anything else? So they're really like honing in on that. And I think, yeah, we don't really know that at this point, but that would be a great research question.
Okay, let's give Stephanie one last round of applause. Okay, next up we have Reed Kenny. Reed is a PhD student at UC Davis, where he's working with Dan Potter to understand the evolutionary relationships among juncus. That's what he's going to talk about today with us. He is a former master's student from Cal Poly in San Luis Obispo, where he did a floristic study of the Swanton Pacific Ranch right before it burned up. So those specimens will be very valuable for us going forward. So let's give Reed Kenny a round of applause and welcome him up to the stage.
All right, yeah, it's so good to be here in person again. CNPS was my very first conference I went to as an undergrad, so it's pretty cool to be up here and tell you all about how I'm going to split juncus into 7 different genera, which you all need to identify in the future. Yeah, just totally joking about that, don't worry. Yeah, let's get going. So just brief overview. I'll say a few words about juncus, why it's such a cool group, and you should all love it as much as I do. Introduce sort of a taxonomic puzzle in the genus and talk about some previous hypotheses as to the resolutions, ideas about that. Quickly, some methods that I've done to look into these questions, and then give you some preliminary results. But yeah, before we get into that, just wanted to say a brief word about juncus. So here's a variety of species, and I think people don't necessarily realize how diverse a group this really is. So for those of you who aren't familiar, juncus is a group of primarily perennial plants that are generally moisture loving, so found in wetlands and riparian areas. However, there's actually annuals in this group, and these plants are found in a very wide diversity of habitats. So you can see here we have some coastal dunes. These are common in salt marshes, but then also you can find them in the desert, in these oases, alkali flats, all the way up to the alpine zone, and also in stuff like vernal pools and damp sand and gravel. So these are actually occupying this really wide diversity of niches. And they can also be, I think, a lot more morphologically diverse than we might think. Maybe you think of a brush as just sort of this boring grass-like plant, which I'll admit a lot of them are sometimes, but there are also these beautiful forms where they can have multicolored tepals, a wide variety of inflorescence morphologies. Many of them have these glorious pink stigmas that if you catch them at the right time, you can enjoy. And my favorite juncus morphology is actually represented on here. There's this little juncus off towards your right here. If you look at the stamens, they make these really cool little soft serve ice cream cones. And I don't know why some species do that, but I really enjoy it. Yeah, so that's just to say a little bit about why I think this is a really interesting group in which to study adaptation and evolution of different morphologies. And juncus has also been relatively understudied in terms of understanding the real evolutionary relationships here. So this is my PhD work, and I'm really just getting going, but I'll talk to you a little bit about what I'm working on. So sort of the big puzzle that drew me into juncus, besides just how great they are, is that the first molecular work that was done in this group uncovered this very interesting pattern of several South American genera in Juncaceae that appear to be very closely related to juncus species. So here I have a couple phylogenies, which if you're not familiar, is just sort of like a family tree for evolutionary relatedness. And these orange rectangles here are highlighting these South American genera. So these are not named as juncus, but if you look at this phylogeny, essentially you can see that some juncus are more related to these genera than they are to the rest of the genus Juncus. So that poses a taxonomic issue for us as taxonomists. These days, we want our groups to all share a closest common ancestor. So this has not been resolved. We've known this for a little bit, but we need, we don't want to make any rash moves here, so we need to get some more information. And So this was something that's very interesting to me. And essentially the big question here is, what are these juncus species that are so closely related to these South American plants? And this result is so puzzling, right? Why we would not have expected this is because these South American genera look really different from the rest of juncus. So that's a photo in the middle of a zoom in on one of these South American species. And I'll just note here for the rest of the talk, if you see SSA genera, it means small South American genera. They're not actually small plants, but there's not very many species in those genera. So small South American genera. So this is what they look like. This is a zoom out. Most of the species in this group are these alpine cushion plants. And yeah, you can see you would not peg this as a juncus if you were just walking around. At least I wouldn't. And you might be wondering, how could this possibly be so closely related to the rest of Juncaceae if they look so different? I don't know the answer to that, but one possible explanation, if perhaps you're familiar with the Hawaiian silver swords, these are some amazing plants in Hawaii that are actually closely related to our California tarweeds. They look extremely different, but we do know that in some circumstances when a species colonizes a new area, you can have rapid morphological change to adapt to a new habitat, which is something that we're interested in as evolutionary biologists, how that happens. So this is something that I saw and became very interested in answering this question. Is this one potential scenario for why we have these cushion plants being closely related to the rest of juncus? And just a quick note, another feature of these plants is that they have solitary inflorescences. That's how the original workers in this group sort of grouped things out and said, well, aside from the cushion plant thing, they're definitely not closely related because all juncus tend to have these flowers that occur in groups instead of singly. So we can also, that's nice. So my colored squares didn't show up here, which is very strange. But I'll just paint you a picture with words. So these species are occurring on the high elevations in the Andes here. So basically ascending down through Chile, all the way down through the tip of South America. And interesting note, it's unrelated to the rest of my research. They're not going to talk about it today, but you actually have some of these species occurring at the tip of South America and also in New Zealand and a couple sub-Antarctic islands. So just to tell you about some other cool puzzles in this group. So I mentioned we're very interested in, okay, what are these species potentially very closely related to? And sort of the preliminary results from those original phylogenies indicate that there's two groups here. And so I'm going to take a second to introduce those other two groups. The first has been named as section Cespitosi. So that is a group of these small annual plants. These are actually highly diverse in California. So California is their hotspot of diversity. Although they occur up to Washington and interior to the Rockies, some species. These guys are really small. You can see that photo with the pencil for reference. That's about 1 centimeter tall. And they're also actually rather divergent for the genus Juncus. So they're annual. I mentioned most Juncus are perennial. And some of them only have 4 tepals instead of the usual 6, which the number of floral parts in a group is very indicative. So those are some interesting differences and sort of indicate, okay, why maybe these are more similar to those South American plants. And oh, great. Okay, here we can see the range of these species. So you can see there's the hot spot in California here, and then the other really interesting distribution here is that these are also found in South Africa, or plants that have been placed into this group are found in South Africa. So these are also annual plants, and South Africa shares our Mediterranean climate. So it sort of makes sense why we might see the same kinds of adaptations, but the distribution here is obviously very unusual, so we'll talk a little bit more about that. The other group that's very interesting here, and that comes out in the phylogeny as potentially the closest group to these South American genera is this section Graminifoli. These are the grass-leaved juncus. So just like I was saying, juncus is actually very diverse. Not all juncus have the typical round leaves you might associate with them. And so these grass-leaf juncus are generally defined by the presence of these flat grass-like leaves. This is one of our California native species, Juncus orthophilus here. And, oh, dang. Okay, well, these have a very cool distribution, which, I'll briefly just go over it. They also have a hotspot of diversity in California and North America in general, but they members that have species placed into this group are also found in South America. They're also found in South Africa, and then they have another sort of center of diversity in southeastern Australia and New Zealand. So we're starting to see some alignment in potential distributional patterns here. So with that, I will get to some previous work in this group. So Barbara Erder was a member of our California botanical community, and she actually monographed the annual juncus of Western North America for her PhD work back in the 80s. And she had no reason to think that these small South American genera were closer related to section Cespitosi or section Graminifoli here, but she did do some really good intensive work onto what the closest relatives were in the currently recognized juncus at that time to those groups, and that has been very informative for shaping my hypotheses as well. So I wanted to go over some of her thoughts here. And her sort of first hypothesis I want to introduce is that she took a really close look at these plants in Western North America and South Africa and said, this seems very unlikely that these are actually each other's closest relatives. If you look closely, these plants in South Africa are really not that similar to Western North America, plus the distributional issue, she said, I don't think these should actually be in the same group. So that's her first hypothesis. Second hypothesis, she more broadly tried to conclude what are the probable most common ancestor of these annual juncus in Western North America. And really interestingly, she actually concluded that there was the South American grass-leaved juncus. When you looked at their seed characters and other really minute morphological details, she actually concluded these are more similar to our North American annuals, which is a really interesting conclusion, and also gets at this North American, South American divide. And so I've just sort of made a simple phylogeny illustrating what her hypotheses based off of morphology were here. And so I really based my hypotheses off of hers and said, okay, well, can I go test this with molecular data? And also, knowing what I know about the likely placement of these small South American genera, can I ask where those are also going to fall out in this phylogeny? So I rode my bike around California and collected a lot of tiny, tiny juncus, as well as a lot of grass-leaf juncus, and I extracted their DNA and sequenced some of it, and also downloaded a lot of sequences from GenBank, which had not been placed on those previous phylogenies. And I made a tree. I built a tree, right, to start and answer these questions. Yeah, that'll do. So there's some points missing on this as well, but this is perfectly good. So what does this tell us? So these are my preliminary results here, and if we start at the top of this phylogeny, so you can see there's colored boxes at the tip of each species there. Those colored boxes are color-coded by what taxonomic section those species had been assigned to. So if we look at the top of this phylogeny outlined in that orange box, you can see what this is telling us is that North American annuals in sort of light orange there are coming out very closely related to North American grass leaf juncus in red. So this is not supporting Barbara Erder's conclusion based off of morphology, but it does make a lot of geographical sense in that these two groups occur very closely together. It makes sense they're closely related. And it does lend some support to our other hypothesis, though, if the closest relative on the phylogeny that we see for these North American annuals are the North American grass leaf juncus, it seems very unlikely that we're going to find that the South American annuals are actually very close related to our South African annuals are very close related to our North American annuals. So supports one of her hypotheses. Unfortunately, I don't have any sequence data from any of these South African annuals. That's something I'm working on getting. And so can't say conclusively these are different groups, but I definitely anticipate that we will find these are not closely related. So the other thing we can see on this phylogeny is that we do have data for these small South American genera here. See that red line pointing to the Andes. And interestingly, what this is telling us is that these small South American genera are actually most closely related to the grass leaf juncus that we find in South Africa, which was a very surprising result. So in this purple box that's outlining the South African grass leaf juncus, there is actually one species of juncus represented in there that is a South American grass leaf juncus. It's actually found in Chile and New Zealand, so it's got an interesting distribution. But it looks like there is sort of a North American group here and a Southern Hemisphere group happening, which there's a lot more to be delved into here, but I thought these were some interesting results and interesting to see what was supported by previous morphological work and what has come out to look different. And yeah, I'll just wrap up with that. Leave you on this simplified slide here so you can see just the relationships between groups. So South African Graminifolia closely related to South African Cespitosi. That's sort of a current hypothesis. And then we can see these small South American genera coming out as closely related to those groups as well. Then we have our North American juncus as sort of forming their own group. So yeah, that's what I've been doing. And I'll just say a note about some future work. I do have plans to add a lot more data to look at this phylogeny, look at a lot more species, and hopefully build something that we can really confidently say these are the relationships here, because the preliminary results, we want to be really sure before, say, we make any taxonomic revisions or publish something that people will use in future research. And I also am really curious about these biogeographic patterns. I mentioned a couple of these interesting groups that are related between continents, stuff you wouldn't necessarily anticipate. So I want to try and solve some of those mysteries. And then, yeah, also work on Western North American species delimitation. We all know there's some very puzzling things in juncus, and I do want to bring hopefully some light to those. So I have Juncus saxmontanus and Juncus encephalius up here as a species pair that could use some clarification in North America, and hopefully molecular tools will aid in that. So with that, I'd just like to say thank you to everybody that's provided funding for this work, and my lab at UC Davis, and you all for being here.
There's time for one question.
Great talk, Reed. I was just wondering, what do we know about juncus's ability to disperse? Like, are they good at dispersing or not very good?
Yeah, that's a great question. That's actually definitely want to do some work on that for my dissertation. It's unknown, but we know there are species that are globally distributed. So, sort of inferring from that, you would think these are able to get around. It's interesting, though, because their seeds don't have any of the sort of characteristic seed dispersal traits that we imagine for really widely distributed seed species. So their seeds are small and smooth. They don't have hooks or barbs. The one thing that I'm really interested in looking into is that some of the seeds form a mucilaginous seed coat, like a chia seed might. And I think that could be a potential dispersal mechanism, in that if you're like a duck or a waterfowl or something in there eating juncus seeds, if it's mucilaginous, it seems much more likely to stick to your beak or your foot, and that could actually disperse along a migratory pathway. So that's something I'm going to be looking into with those biogeographic analyses I want to do.
Okay, let's give Reed one last round of applause.
Next up, we have Adriana Hernandez. She is a postdoc at the Cal Academy, and she is working with Sarah Jones on Castanea. But today she's going to talk a little bit about some of the work that she did for her PhD, looking at the evolution and flora morphology in Calochortus. So let's welcome up Adriana.
From Calochortus to Castanea. Good morning, everyone. Thank you for coming, for being with us today on the last day of the conference. We're all a little tired. Today I'm going to talk about how we used RADseq data from a population genetics study, which you may be familiar with, to leverage that same data through genome-wide association studies and identify loci that are under selection from climate. So one of the biggest questions of our time is how populations are responding and will respond to climate change. One way we can begin to get at this is to look in the past. How have populations responded to and adapted to climate across broad geographic scales? I'm trying to get the pointer going. Great. So one way to assess this is looking at phenotypic differences through reciprocal transplant and common garden experiments. People have also begun to look at natural history collections, which capture phenological differences across time and across broad geographic scales and climate regions. But what about the genotype? What do we expect? What kind of genotypic changes do we expect across various climate? So we expect loci under climate selection to show allelic variation in response to different climates. What do I mean by that? So alleles under selection from climate are expected to track environmental clines. I have one example here on the left that shows two discrete environments, environment in orange, environment blue. They're very discrete. We see a fitness optimum. This is not higher or lower fitness, but the fitness optimum for one environment, and then a sharp cline into a fitness optimum for a different environment. So the traits that are associated with that fitness and the allele frequencies that are associated with that fitness are also then following this sharp cline. What about an environment with a softer gradient? Well, we expect the fitness, the fitness optimum for that gradient across environments to also follow a steady cline. And so then the trait values for that associated with that fitness and allele frequencies associated with that fitness optimum to also then follow these steady clines across geography or steady environmental gradients. However, a lot of this is theoretical. There is empirical and theoretical evidence lacking for this hypothesis. What has been studied are often model organisms, so Arabidopsis, North American house mice, Drosophila. So I encourage all of you, if you have RADseq data across clines, to leverage that to study these environmental gradients, environmental clines. So what's the study system? Calochortus venustus. I don't know why this is coming out weird. So all of this diversity that you see here is one species, Calochortus venustus, the butterfly Mariposa lily. We see variation in flower color from white to yellow, purples, pinks, and reds. There's variation in 4 other floral traits as well, which I don't have time to talk about today. but I'm happy to talk about after. And we summarize these floral phenotypes across California. This is a California endemic. So I have the Central Valley here with our samples in these deep blue triangles. And these collections were based from herbaria specimens, which are in the light blue dots. And what we see is that the southern populations are the most and highly polymorphic, with every color represented. But along this cline going north in the Coast Ranges, we see less and less pigmentation and almost exclusively monomorphically white populations in the north going up to Mount Diablo. Similarly, along the Sierra Nevada, we see less and less color up to the point where we see almost exclusively monomorphically white populations in Stanislaus. So we see parallel clinal patterns of floral pigmentation. I then used RADseq data, 94,000 SNPs across 174 individuals, and we found gene flow between all floral phenotypes and between populations. We also found a stepping stone model of population structure. Essentially, individuals that are most geographically close are also genetically related, genetically close. And we can even see in this structure plot this kind of steady gradient, somewhat of a steady gradient across populations. These are the coastal populations. And then we see the transverse individuals going into the Sierra Nevada, with less and less genotypic similarity going into the northern Sierra. So we also used historical range reconstruction to figure out past biogeographical events and found a bidirectional south to north migration, or range expansion, where individuals here in the Carrizo Plain appear to have moved north into the Mount Diablo region and from the transverse ranges across into the Sierra Nevada and also going north. So we found, in addition to parallel morphological convergence, this parallel historical expansion. So back to our question, how have populations responded to and adapted to climate across broad geographic scales? And again, we expect the loci that are under selection to show clinal, these allele frequencies to be steadily clinal in response to clinal climate. So we wanted to know if allelic clines tracked clinal environments in Calochortus fenestis. And given that we have these two transects, we wondered if the expansion of the range expansion from southern to northern habitats, which are similar in many ways, would show parallel allelic variation. So in other words, do our two transects show the same allelic similarity for some SNPs under selection? And we also know that the Sierra Nevada and coastal populations are also very different in many ways, right? Sierra snowpack, coastal populations undergo a little bit more of a temperate regime and also deal with things like salt and high winds. And so we wondered if the climate pressures that show longitudinal clines, in other words, differences between one longitude and the other extreme, would impose divergent selection pressures between our two transects. So really we imagine that we would find both parallel allelic signatures of selection as well as divergent signatures. So we identified SNPs under selection from climate, first by identifying climate variables that are likely to impose spatially varying selection across our samples. These 9 climatic variables are known to influence growth, phenology, and distribution in other California native flora. And we made sure that they reflected latitudinal or longitudinal or both clines along the distribution of our 174 individuals. And again, we leveraged that same RADseq data for the PopGen to find statistically significant associations between our RADseq SNPs and the 9 climatic variables, in other words, GWAS. This is a special type of GWAS called latent factor mixed models, which incorporate population structure, given that we have very strong population structure in our system. So we started with the 94,000 SNPs across 174 individuals and ended up with 3,700 SNPs that were uniquely associated with one of our 9 climate variables. We did find SNPs that were associated with multiple climate variables, but then it's difficult to tease apart what that climate variable, what the contribution of each climate variable is on that SNP. So for these 9,700 SNPs, we measured clinal variation in allele frequencies, expecting to find those nice steady clines. What does it look like when we're not looking at a SNP under selection? So we took from the 94,000 SNP data set a random 1,000 SNPs, and we looked at allele frequencies for populations in the coast and allele frequencies of populations inland in the Sierra Nevada. So on the X here is latitude, and on the Y-axis are allele frequencies. So this is the proportion of individuals in a population that show a specific allele. So here we see that at this SNP, the allele is essentially fixed. 100% of individuals have this allele, whereas down here, 100% of individuals have a different allele. You can imagine this as like A or G, for example. And so we looked at clinal variation again in both transects, and no SNPs showed parallel allele frequencies, parallel clines in both transects. A small percent did show clinal variation in maybe one transect, but not both. And this is what we expect, right? These are 1,000 just random SNPs that we don't expect to be under selection. So we don't see clinal variation. So what about the SNPs that we do think are under selection, that are strongly associated with climate variables? So I have mean annual temperature up here, again, across latitude, and we see higher temperatures at one end, the Sierra Nevada, and lower temperatures, actually more variation, in the south, this is the north. And the climate variables that showed the strongest cline, so the strongest R value, were also associated with the highest number of SNPs that showed parallel clinal variation. So here I have one example of a SNP. This is the chalcone synthase gene in the coastal transect that shows this clinal variation, as well as in the inland transect in the Sierra Nevada. So if they both had an R squared value, an R value of over 0.4, we would say that they're both showing parallel signatures. So these parallel clines and allele frequencies strongly support the hypothesis that the SNPs that we identified are indeed under selection. What about longitudinal clines? So we sampled again 1,000 random SNPs and looked at allele frequencies across longitude. So now this is coastal, and to the right are Sierra Nevada. And only 8% showed clinal variation in allele frequencies along longitude, right? So this is just very random here, as we would expect from a random 1,000 SNPs. So longitudinal variation here, this is minimum temperature of the coldest month. So we see that the minimum temperature in the coastal populations is much higher than the minimum temperature in the Sierra Nevada, which again we would expect with that Sierra snowpack. But we do see more variation in the Sierran populations due to that complex terrain, right? And again, the climate variables that show the strongest longitudinal cline also are associated with the highest percent of SNPs that show clinal variation with latitude. So this is one example, the CBL chitase 8 gene, which regulates salt tolerance. And we see these allele frequencies are not as broad. There isn't as much variation in allele frequencies for the coastal population, which we would expect because those are undergoing likely more salt tolerance. So the climate variables exhibiting longitudinal clines seem to be driving selection, leading to the divergence of our two transects in the Coast Ranges and the Sierra Nevada. So do allelic clines track clinal climate pressures in Calochortus venustus? And the answer is yes. We found empirical evidence that alleles under selection track these environmental clines, and that the strength of the allelic cline for SNPs under selection seems to depend on the strength of selection. In other words, those climate variables that show stronger clines are associated with more SNPs that also show allelic clinal variation. What this shows is that studying the effects of climate on one population cannot be used to predict the response of other populations, which may be undergoing and likely do undergo different climate pressures, especially if they have broad geographic ranges, distributions. So populations under different climate regimes do respond differently, and we find evidence for that at the molecular level. And we identified allelic clinal variation as the genetic mechanism that underpins that population's specific response to climate. Allelic clinal variations suggest clinal adaptation to climate, that these identified SNPs are under selection, that molecular evolution can occur in parallel. In other words, that we see the same clines for some SNPs in both of our transects, which undergo similar environments. And climate selection is continuous along these clines, right, as we see these steady clines and not discrete clines. So we do indeed find evidence for both parallel and divergent genetic evolution of these adaptive alleles in the face of ongoing gene flow. Why is that important? So these parallel genome evolution, in other words, parallel allelic lines, could be facilitated by recruitment of standing genetic variation, given that this is all one species, so that allele could have been inherited, but also because of gene flow here through the transverse ranges. The transverse ranges are important in connecting our two transects, and this is, perhaps not coincidentally, also a region of high phenotypic diversity. Divergent molecular evolution between our two transects, coastal inland lineages, was achieved despite higher rates of gene flow, suggesting that the strength of gene flow, the strength of selection, or that climate pressure, counteracted gene flow. Not sure how I'm doing on time. Okay, great, right on time. So I don't have too much time to talk about this today, but what are these SNPs doing? What are they, right? So this is a non-model organism, but we can use known functions from other model organisms like rice and Arabidopsis and so on to look for sequence similarity. I then characterized the known functions of those similar sequences in other taxa to look for key functions like floral phenotype, looked for petal, anthocyanin, trichome, and symmetry, for example, given that we know these traits are geographically structured. However, we only found 119 of the 3,700 SNPs to be associated with floral phenotype. We found a much higher proportion of SNPs to be associated with root, leaf, cotyledon, pollen, germination, and even more associated with California climate, cold, freezing heat, drought, fire, and salt stress, for example. And this makes sense because the way we identified these loci are through climate variables, so it makes sense that we find they're associated with cold, freezing heat, drought, et cetera, right? And these plant and bulb functions take environmental cues. So again, it makes sense that we have this finding. With that, I just want to thank the Spec Lab and everyone at Cornell who helped push this project forward, as well as land managers through the UC Natural Reserve System. I see Zach in the audience. And the Santa Barbara Botanic Garden, who was kind of a home base for me when I was doing some lab work and field work here. My funding sources. Thanks.
Questions?
Hi, thanks for your talk. Do you have an idea about the time frame under which this variation occurred? And could you speculate about how that might inform our ideas about adaptation to climate change?
That is a great question. How can we apply this kind of data, right? Well, we don't have a time-calibrated tree for Calochortus yet. There are no fossils. We estimate that the lineage is maybe 7 million years old, which is fairly young for plants, but also that's 7 million years, right? And we're thinking about climate change in 100 years or now. So I don't have a good answer for you, but that's where this kind of work gives us a kind of jumping point to do those common garden experiments, given that we know what to expect in differences in allele frequencies, we know what climate variables are important for specific populations. And so I'd say this work definitely is just a springboard for future experiments.
Other questions? I have a quick one. To me, your big results sort of suggest that there's local adaptation. Like that's the framework I think of this in. It's like there's been local adaptation to climate on these parallel transects, essentially. And I'm just wondering if you've thought of like, what are the implications for that in terms of thinking of like moving seeds and response to climate in the future and restoration? I don't know. Just any thoughts on that?
Yeah. So I would say, fortunately, Calochortus vanensis is not a rare or threatened, I would say, because there are thousands of individuals, even though they are being poached. was kind of afraid to like post my coordinates for my study system, right? Because I mean they're beautiful, so can you blame them? But yes, we can. They should not be poaching plants. No, so I would say that we have to do those common garden experiments, those reciprocal transplants, to kind of solidify. For one, to link the genotype back to the phenotype. So we know what's happening genetically, but what is the function of that, the ultimate, kind of ecological function? So there's a kind of big in between getting this genotypic information and moving forward with any kind of, restoration work.
Thank you.
Yeah.
Okay, next up we have Ioana Anghel. She's a PhD student at UCLA, and she is working with Felipe Zapata on the diversification in the genus Linanthus, which is a really awesome clade of plants full of color diversity and mating system diversity and different soil endemisms. So please welcome up Ioana.
Hi, everyone. Hello. Nice to see you all. Thank you for being here. I'm Ioana. I'm from UCLA. You don't hear an echo, right? I'm hearing some echo. Sounds good? Okay. So I'm going to talk to you today about my PhD work. I work in the genus Linanthus, and I'm going to talk to you about why it's so cool and so interesting and some of the questions that I'm trying to explore there. So, as you all know, this is why we're all here. California is an amazing place with a lot of biodiversity. It's basically a hotspot for plant biodiversity, with many lineages that are diversifying in this region. The Mojave Desert, especially, and some of the areas around it, seem to be a cradle of young plant diversity in California. Looking at this this map from a paper by Nathan Kraft et al., they show that a lot of young endemics are occurring in this area, and I'm going to try to use the pointer. A lot of young endemics are occurring in this red area. So basically, Mojave Desert and some of the transverse ranges, you can see there's a lot of young species there. So it seems like the desert is sort of acting as a pump of new species in California. Another special thing about California flora is it seems like a lot of sister species co-occur. This is a paper by Anna Strauss where they looked at basically the age of sister species on the x-axis and how much these species overlapped in space on the y-axis. And you can see that there's a lot more of the younger sister species that are basically overlapping much more. So this has implications for how plants are occurring in space and how they may have diverged with a higher opportunity for gene flow. And so how have so many species evolved here despite this homogenizing effect of gene flow? And so I'm interested in this genus Linanthus, and it's a very cool system for understanding these questions about evolution because it's a young, diverse, and arid-adapted genus with a high range overlap. As you can see here, there's some examples of the diversity in the group, but there's 25 species with a lot of them occurring in Southern California, so kind of a hot spot in Southern California, in some of those areas that I showed you in the Mojave Desert, as well as the transverse ranges. So the species also has a lot of color and morphological diversity, as you can see here, with many species having white, yellows, lavenders, pinks, purples, and indigo flower color. We also see a lot of throat and tube marking diversity between species and within species. And you can see here some of the most variable are Linanthus dianthiflorus that you may have seen on the coast in Southern California, this one here, and Linanthus perry that I think a lot of you might be familiar with down here. Those are some of the most variable ones in terms of tube markings. We also have a lot of really cool diversity in flower opening time. These species here on your left side are flowers that open at night, while the ones on the right are some of the examples of the flowers that open during the day. So another cool dimension of diversity in this group. And just to kind of define these terms, vespertine refers to night opening, and diurnal refers to the day opening flowers. We also see a wide range of scent produced in the flowers, which is exciting to be in the field and sniff these and try to describe what they smell like. So here's a little sommelier's guide to Linanthus. We have Linanthus inuensis, which my friend Sarah Jacobs, actually Adriana's new advisor, described as that it smells like bad breath and mothballs, which I love. It's very vivid. Linanthus dichotomous is one of the night opening ones, and it smells a little musky and like lilac. Linanthus maculatus over here, it's a very small plant. It smells a little putrid and a bit intoxicating. And Linanthus perrier, the one you all may be very familiar with, it smells like grape jelly bean, at least the blue version. The white one smells a little different to me. And so it's interesting because scent can be kind of it's been shown that in some cases it can be a mechanism of reproductive isolation, which may be somehow how these plants are diverging, even though they're locally co-occurring. And a lot of different insects are visiting the different species. We see flies or beetles or hawk moths or sometimes long-tongued flies visiting them. And we also see a lot of within species diversity. And the famous one, again, is Linanthus perrier, but we see this in actually a lot of other species. So like Linanthus orchidii, Dianthiflorus callipii, a lot of these have within population variation as well in color. So polymorphisms are really interesting because they can help us understand how this multiple forms are maintained within populations, even though there is gene flow. So how are they maintaining this phenotypic homogeneity? So just to show you kind of where they occur in California, at least, another reason why this group is interesting is that there's a lot of range overlap. So these species, we think they're about 7 million years old, the group, but there's a lot of range overlap in many of these closely related species, kind of in the same areas that I was mentioning before, Mojave transverse ranges, a little bit, mostly Southern California. This is quantification of range overlap, and basically you have sister species, not sister species, but species pairs matched in this graph, and you can see that the darker boxes represent areas that are, or species that have high range overlap. So lots of species have some overlap in their range, in their distribution. But also, it's really cool seeing them in the field. I'm seeing a lot of different species that are super locally co-occurring. So over here you see Linanthus dichotomous and maculatus down here, as well as, I know this is a bit hard to see, but this is Linanthus demyscis and Linanthus filiformis down here, this yellow flowered species, where they're basically, you find one and you find another one right next to it. So it's really cool to see that they're very, they occupy the same niche, it seems like, at least soil-wise. And so it's interesting to see these really closely related species occurring in such basically small space, because sometimes species that are closely related may not have complete reproductive isolation. So there may be an opportunity for gene flow. So all of these interesting aspects of Linanthus diversity make it such an interesting system to study questions about how diversity is created and maintained, especially in the context of that it's a young genus that has rapidly diversified, occurring in these arid environments that are basically very harsh, so you have to reproduce and live in a very quick amount of time, with lots of polymorphisms, as I've showed you, and lots of sympatry and overlapping ranges. So to first answer these really interesting questions about how these processes of evolution might work, the first step is basically building a good phylogeny. So that's what I'm going to talk to you about today. So there have been some There have been some phylogenies built for Linanthus before. This is one of the earlier attempts with ITS data. This is from a paper by Bell, Patterson, and Hamilton. And this is kind of the first paper that showed the monophyly of the genus. You can see that there's some old names for some of the taxa, but basically this is showing us that a lot of these species are monophyletic, actually, and they're all in Linanthus. And so this is not actually including all the species. In another attempt from 2017 by Jacob Landis, he used target capture data. And I think he used about 1 to 4 individuals per species. So the relationships become much more clear here. But as you can see, there's still some questions about certain species. For example, Linanthus filiformis is in green is falling up here with Campanulatus, but also down here with Jonesii. So to kind of make this, to try to make this phylogeny more robust and try to include a lot more individuals, this is kind of my process. So I collected samples in the field and also supplemented them with herbaria specimens. I extracted DNA from these samples, prepared libraries for sequencing and assembly, and then did some analysis. So I used 192 samples. A third of them were field collected and 2/3 from herbaria. Here is kind of the distribution of all the samples that I used in the study. The different colors represent the 25 different species that I included, which are all the recognized taxa, with about 7 samples per species. And I also used a lot of individuals that were locally sympatric that were collected from the same place. Just to show you some of the fun you can have with liquid nitrogen. This is, so some of the samples were collected in liquid nitrogen in the field and also in silica. And traveled a lot across the states at 112 populations. And then with a lot of generous contributions from herbaria, I was able to supplement my sampling. So then we did extractions with the help of 2 undergrads, Jade Corpus Sapida and Sarah Payne. And this is just to show you some of the tissue ground up. It looks kind of cool with all the different colors depending on how much petal tissue you use. And prepared libraries through RAD-seq, which is kind of a method to cut the DNA across many different sites across the genome, and it's much more cost-effective than doing whole genome and the entire genome. So then I assembled those loci using IPyRAD. And map those reads to a new reference genome that we actually just published in September on the Linanthus berriae, so that's very exciting. So that helped me get a lot more loci after we actually had that genome. So after filtering, I was able to keep 180 of those samples and use those loci to build an IQ tree phylogeny. Basically, it's just a program that is kind of fast and widely used to infer a phylogenetic tree. And it's a maximum likelihood method, so using those rad loci. This is a terrible picture, but I just wanted to show you what they all look like together. So it's a lot of samples in this phylogeny. To kind of simplify that for us, I collapsed some of the clades. So basically, the way it's color coordinated here, you can see the branches in green, and the tips in green are species that were monophyletic. So basically, all of the samples in that species fell into the same branch. And so, as I was mentioning, there's about like 5 to 15 individuals in each of those groups, and it's quite exciting when they actually all look like they're each other's closest relative. So Linanthus campanulatus, filiformis, inuensis, arenicola, berriae, demysis, Danthiflorus, and maculatus were all monophyletic. So that's exciting. But then the other colors represent, the yellow is basically clades that were mostly monophyletic, but with one or two intruders. And so potentially there may have been some misidentification there. And then the pinks are these 2 clades. One of them is this Vespertine group that I was mentioning, the night opening one, with Linanthus dichotomous, Biglovia, and Jonesii, that had a lot of confusion. And I'm going to focus in on some of these. Then this is the perennial clade that also did not seem to be, the species were not monophyletic. To kind of explain where these clades fell into geography. A map would have been helpful here, but I'll just explain this. Basically, one group is kind of Owens Valley, Eastern Sierra, Great Basin. We have another group in the SoCal deserts. Then we have the woody species that are coming out here, the coastal California and Baja species, then Eastern California and transverse ranges, and then these night blooming ones that are monophyletic clade. Now I'm going to focus on these perennial species that are these basically woody perennial species that I mentioned here. Again, all the samples, but we don't want to look at that because it's too much information. And as you can see here, just when we believe the labels of these species, it's very much a mess. You might be familiar with Linanthus pungens from California, and Linanthus californicus as well, and maybe Jägeri. Californicus kind of occurs on the coast in California, pungens sort of anywhere in the mountains, but mostly in the Sierras and the transverse ranges. And Jägeri is San Bernardino mountains endemic. And it's interesting that Jägeri is coming out both here and here. Does not seem to be monophyletic, so that's interesting. One big thing about this is that the perennial habitat, these collapsed clades are all the other clades in the genus that are annual, actually. So this perennial clade seems to have evolved from this annual habit. And to zoom in on one of these clades, Linanthus cespitosus, watsonii, and glabris are also in the same order in these maps here and with the images. As you can see, these are the most widespread species in the genus, and they sort of overlap in space, and their relationships are very much not clear. So there's a lot more work that needs to be done here. Then to focus on another example, which is this vespertine night opening species. Linanthus dichotomous is here on the left, and it's kind of one that has a lot bigger flowers and this pinkish hue on the outside of the petals. In the middle, we have Linanthus biglovii, which basically looks like a smaller Linanthus dichotomous, and Linanthus jonesii, which is a species that has these glandular trichomes as well as a more yellowish petal. So here I used basically 11 species of jonesii and biglovii and 17 of dichotomous. And again, this is kind of a huge mess. Linanthus biglovii and jonesii, there's a clade here represented in yellow. They're kind of all over the distribution of all of these species, as well as another clade here with Linanthus dichotomous and jonesii in this purple. And then it's just basically Linanthus biglovii comes out in this clade as well, and dichotomous again in this clade in blue. So it just seems like there's no real geographic or species structure at this point. What might cause these patterns, you might ask? It might be maybe taxonomic misassignment, or people didn't know which one was which, perhaps because some of them look similar. But also, maybe this is an example where gene flow or incipient speciation is occurring. This is one area of focus for these questions of how these species have evolved and how does that happen within this context. And another cool example is these local endemic species called Linanthus callipii and bernardinus. This is what they look like. On the left, you see bernardinus. It's kind of basically they look quite similar, but Linanthus bernardinus has a longer tube and a taller flower, while Linanthus callipii, these are all of the variations of this species. You can see petals that are white, petals that are purple, sometimes a little like in between, like pinkish, and then the throats are sometimes yellow, sometimes white or purple, and all of the combinations in between, which is super cool. There's a wide morphological diversity here. They occur in the San Bernardino mountain area. Callipii, I don't know if you're familiar with San Bernardinos, but Big Bear is over here. And this area around Baldwin Lake is where Linanthus callipii is historically known to be from. So the yellows are this Linanthus callipii. And then in green, you can see kind of the historically known populations of Linanthus bernardinus. In pink here are kind of this new, maybe perhaps transition population between the two that are, I've called them Loanthus callipii here, but we're not really sure which species they are at this point. But you can see in the phylogeny, Loanthus bernardinus is nested within these samples of Loanthus callipii, and also this population in the middle seems to not be monophyletic, as well as this yellow, originally thought to be the Loanthus callipii area. So just to conclude everything I've talked about, many species are monophyletic, which is exciting. It kind of confirms that perhaps the phylogeny looks good, and it's great. Species that are monophyletic are great. And then we have the few problem children, which is the perennial clade, which is monophyletic itself, but the species in the clade are not, and it evolved from this annual habit. The night blooming clade needs a lot of work, perhaps due to some taxonomic work that needs to be done, or gene flow, or speciation. Then this Loanthus bernardinus being nested within Loanthus callipii, a lot of work needs to be done to figure out how exactly those populations are structured. So in the future, I'm going to be doing a lot more work trying to basically look at these population structure in these examples that I've given you, and investigate admixture patterns between species. And then I'm also, part of my project is also looking at floral volatiles in the genus. So that's the part I'm very excited about that I mentioned earlier. And again, that's interesting because in a lot of species co-occur. This example here is from Shell Creek in San Luis Obispo County. I was taking scent from Loanthus dichotomous that was sympatric with the Loanthus perrier. And also, I'm exploring landscape genomics of Loanthus, the two sister species, Loanthus perrier, that you're all familiar with Loanthus callipii, from 150 populations across their entire range to understand processes behind speciation. Thank you all so much for listening, and especially I want to thank the California Native Plant Society LA Santa Monica Mountains chapter, because they helped me attend this conference. Thank you all so much for listening.
We are out of time for questions, but I'm going to let one go.
Thank you.
Yeah.
So there was many variations of the color of the petals. Have you shone a UV light to look at the nectaries, what actually the pollinators would see? Is there a difference in that to have additional data set?
Oh, sorry. Maybe just do that. Sorry. I have not looked at that, but I would love to see that bring a UV light in the field, especially maybe for some of those night blooming ones. That would be exciting. Yeah, good idea. Thank you.
Thank you.
All right, next up we have Anjum, and she is working at UC Davis with Jennifer Funk on functional trait variation and implications for restoration. So let's please welcome Anjum.
Hi, everyone. Thank you for attending my talk. Like Natalie said, I'm a PhD student at UC Davis, but today I'm going to be presenting on some work that I did in my undergraduate at San Jose State University on the conservation of a rare redwood forest endemic plant, Dudley's Lousewort, and this work was done under the guidance of Dr. Ben Carter and Dr. Tracy Masejewicz, as well as our colleague with the Center for Natural Lands Management, Chris Hauser. So just to start with introducing this plant, Pedicularis dudleyi is really quite a rare plant. We can see from the CNPS rare plant inventory that it has a rare plant designation of 1B.2. And so this means that this plant has likely experienced a steep decline in its numbers over the last century. And we know that there are two extant populations, one at Portola Redwood State Park in the Santa Cruz Mountains, which is where I did much of my research, and one alongside the Little Sur River in San Luis Obispo. And so we're interested in what is causing this plant to be so rare. As plant ecologists, naturalists, conservationists, we're oftentimes interested in conserving biodiversity, and particularly in endemic plants. And so we wanted to understand why this plant is rare in order to better manage and maintain the extant populations. So I started with collecting baseline demographic data for this plant. While we know it's rare, we didn't actually, prior to the study, have numbers to say how many individuals are remaining. And then secondly, we aim to determine whether reproduction and/or regeneration are limiting population growth. And so to get at the first aim of our research, I did a complete census at the Portola Redwood State Park population in 2019. And this consisted of classifying 22 subpopulations into 4 age classes: reproductive adult, a mature but non-reproductive adult, an immature plant, and the seedling stage. And what we found is that there are only less than 500 reproductive individuals at this Portola population. And so the panel on the left indicates our subpopulations that had less than 20 reproductive adults, and the panel on the right indicates our subpopulations with more than 20 reproductive adults. And so we're interested in looking at 5 limits to population persistence. And so these are the potential mechanisms that could be limiting the establishment of new populations, including pollination, seed production, seed dispersal, seedling establishment, and seedling survival. And so in terms of pollination, we first asked what is pollinating this plant, and we were interested in quantifying rates of pollination. And then secondly, whether pollination is even necessary in this plant or whether it can effectively self-pollinate. And so in the bottom right, you can see some pollinator exclusion bags that we use to test this. And what we found is that there are 2 native bumblebees pollinating this plant, Bombus atkensis and Bombus edwardsi. And this had been discovered actually in the 1950s by a woman named Elizabeth Sprague, who studied this plant for her dissertation. And so it's good to see that these plants are still effectively pollinating this plant. And we're able to quantify some rates of visitation. We see that these 2 bees are pollinating at a rate of about 20 flowers per visit to an individual plant, and about 5 to 10 plants per visit to a subpopulation. And so in terms of thinking about mechanisms that might be limiting population persistence, we don't think that pollination is a factor that could be limiting the establishment of new populations. And so the next thing we did was looked at seed production, and we compared naturally pollinated plants to plants in the group with the pollinator exclusion bags on them, and as well as a third group of plants that had been hand pollinated and had pollinators excluded. And what we found is that no fruits matured when pollinators were excluded, but when pollinators are excluded and the flowers were hand pollinated, about 40% of flowers produced mature fruits. And both of these groups are in comparison to our control of our naturally pollinated plants, where we're seeing about 80% of flowers producing mature fruits. And so similar to pollination, we don't think seed production is a factor that could be limiting population persistence. And we were also interested in seed dispersal, and so we used a similar methodology as our pollinator observations with those GoPro cameras to determine what is dispersing the seeds of this plant, and some rates of seed dispersal. And what we found is that wasps, or yellow jackets, are dispersing the seeds, as you can see in this photo with the wasp poking its head into an open fruit capsule. And they're visiting these inflorescences at a rate of about 1 inflorescence an hour. And half of these visits actually result in a wasp flying away with the seed, as you can see in this diagram to the right, by Burge and Beck. And so, interestingly, this has been identified as a mechanism for seed dispersal in California native plants pretty recently. This 2019 paper is the first kind of noted observation of this. And so this plant here is spicebush, Calycanthus occidentalis. And what we also notice is that these wasps aren't necessarily poking their heads into open fruit capsules and pulling seeds out right away, but they actually sometimes hang out with their mandibles in the capsules for sometimes a few minutes. And so we were interested in what these seeds looked like after wasp visitation. And like I mentioned a moment ago, only half of the visits by wasps actually result in a wasp flying away with the seed, but the other half of the time, these seeds remain in the fruit capsules, which allowed us to go back and actually see what these seeds looked like. And so you can see with these microscopic images that the seeds that had been visited very clearly have the outer layer of their seed coating scraped off. And so we don't quite know what this outer layer is. We've speculated that it could be an eliasome, but either way, it seems to be something that's important for attracting seed dispersers. And we can see the figure that when we collected 132 fruits that were opened, about half of their seeds were scraped, whereas the other half were normal. But when we looked at completely closed fruits, 100% of those seeds were normal. And so this suggests that there is this broader mechanism of seed predation happening at this population. And so we are also interested in whether seedling establishment is threatening population persistence, and specifically in the context of some of the seed predation that we're seeing. And so we asked whether germination rates differ between normal and scraped seeds, and whether germination is an issue that we need to think about with establishing new populations. But unfortunately, with our greenhouse experiment, 150 seeds that we had attempted to germinate failed. And so while we're not able to really say at this time whether germination rates differ between the normal and scraped seeds or whether germination is an issue threatening population persistence, we do think that germination seems to be really reliant on the specific field conditions at Portola. But secondly, with thinking about seedling establishment, we noticed observationally that this plant, Pedicularis Dudleyi, doesn't seem to grow in areas of high litter accumulation. And so we asked whether litter accumulation influences seedling establishment. And what we found is that there was no difference between the control and treatment plots of this litter removal experiment, as you can see in these plots. But we think that this experiment was pretty impacted by a low sample size, and so we don't think that we have enough statistical power to completely rule out litter accumulation as a negative, as a factor negatively impacting seedling recruitment. And so we're working with the state parks to actually adapt this protocol to include higher sample size and be able to better understand whether litter accumulation and by extension fire suppression, whether that is negatively affecting seedling establishment. And so while we were limited with the results from our germination and litter removal experiments, we can take a look again at our population census data to kind of extrapolate some rates of germination. And what we found is that there's about 5% of seeds at Portola Redwood State Park that are germinating. But interestingly, the ratio between reproductive adults and seedlings is really quite variable across these subpopulations, which makes it pretty difficult to pinpoint rates of die-off between age classes. But moving forward with rare plant management, we don't think that pollination, seed production, and seed dispersal are factors that are limiting population persistence. But we think that the bottleneck seems to be somewhere in the seed germination, seedling establishment stages. And so we're interested in establishing new populations with outplantings. And fortunately, we know from a naturalist, Kim Kuska, who's been really passionate about studying this plant over the last few decades, that outplantings seem to be successful. Kim planted several individuals in 2001 and 2015, some of which are still alive and reproductive today. And then the second thing that we want to do moving forward is clarify the impact of litter accumulation and fire suppression on the establishment of new populations. And when we went through and did the census in 2019, we put in these metal tags, as you can see with the photo with the banana slug. And so this will allow us to track several hundred individuals through time throughout this population. And so our work has been published. This year we published a paper in Madronio as well as Conservation Genetics. So definitely check those out if you're interested in learning more about our study. And while my talk today focused primarily on the Portola population, Tracy, the co-PI on this project, has been pretty extensively studying the population genetics between the two extant populations. And she gave a talk earlier this morning for anyone that was able to catch it, but definitely check out her paper if you're interested in that little sir population. So with that, I'd just like to acknowledge folks that were pretty crucial to this project, Ben Carter and Tracy, the co-PIs, as well as Chris Hauser with the Center for Natural Lands Management, Ryan Diller with California State Parks, Amy Patton with CNPS, who I think is here. I saw her briefly. Dave Keel, Kim Kuska, Tyler Knapp, and Lars Rosengreen. I'll take any questions.
My mind is just spinning with stuff. Anyway, so interesting on the yellow jacket. Were you able to identify the yellow jacket? Mm-hmm. Was it Pennsylvanica?
The genus is Vespula.
Vespula, is it Pennsylvanica?
I think it starts with an A. I'm blanking on.
Oh, okay. All right. Anyway, did you film it fly off?
Yes, we have video observation of that, yes.
Track the nest.
Great talk, thank you. I'm wondering if you have looked at the role of the wasp in seed germination or removing the lyosome, because ants often collect seeds and they remove the lyosome, or if you tried any scarification treatments in the greenhouse.
Yeah, that's a really good question. So that was actually a component of our germination study. We had I believe it was about half of our seeds were scraped and the other half were normal. And so we were really interested in seeing whether that scraping either promotes or inhibits germination, but we don't have enough information at this time on that, yeah.
I was wondering if you could elaborate on your germination tests and results.
Yeah, so we had 150 seeds that we attempted to germinate in a greenhouse environment at San Jose State. We had several different categories. I think we had a cold stratification, and I can't remember the other ones at this time. I wasn't actually a part of that study, but we collected the soil from Portola, so it had its like native soil conditions. Yeah, does that help answer your question?
I was wondering, is this plant hemiparasitic? And if so, how does that play into its conservation concerns?
Yeah, that's a good question. So I know several individuals that are closely related to this plant are semi-parasitic. We don't have evidence that this plant is, but it could be something that we just don't know yet about.
This family is known not in Pedicularis, and I'm curious to know
what's known about pedicularis, but other Orobanchaceae, the seeds germinate in response to a host exudate of some sort. And I don't even know if pedicularis host is known, if it's a fungus or they attach to a plant or what they're up to under there. Do you know anything for us?
There have been several papers, again, I think from the 1950s, that had kind of explored whether this plant is parasitic, but none, like I said a little earlier, none of those papers actually found evidence for that. Yeah.
Great, thank you so much, and thank you everyone for attending this session. Yeah,
thank you.